Well, ladies and gentlemen, we're back. And the next Stem Cell Emmy Award goes to Dr. Mitch Seyedin, who, believe it or not, I've known since 1995. And every time he starts a company, somebody buys it. He never gives us a chance. But anyway, welcome, Mitch. He's going to talk to you about ISTO technologies. Thank you.
We have a little bit of different company, different technology. It's not exactly stem cells, but we are in regenerative medicine. A little as a way of background about our company, we consider ourselves advanced stage regenerative medicine company. We have platform technologies. We actually two platform technologies, one cell-based, the other one biomaterials. The main focus and what I want to talk today to you about is about cartilage regeneration. We call it, at least in orthopedics, the holy grail of the orthopedics. And as you see for a moment why. And of course, we have a novel biosynthetic, as I said, it's the product that we are marketing today. It's in the market. Very big unmet medical need, and I'm not talking about all the potential procedures, but the number of procedures that we are focusing today with our cartilage regeneration is about over 1 million procedures per year. All right. The three products I just talked to you. First one, we call it RevoFlex. It's the engineered cartilage. We actually believe we're the only company in the world can actually grow cartilage. This is a living cartilage graft. What you're seeing there, someone holding a spatula, a piece of cartilage, is almost like a very large contact lens, transparent. You can almost see through it, and it's focused for sport medicine, knee. I'll expand in a minute. And of course, the second product is called NuQ. That's a minimally invasive cell-based therapy for treatment of degenerative disc diseases, people with the lower back pain. And then finally, the one I'm not going to talk to you today is a biomaterial-based product we're selling. It's called InQ. Again, there we focus mainly in spinal fusion application when there's a tremendous need for bone grafting. Why cartilage? I'm going to again talk about cartilage regeneration. This is an unmet medical need. And keep in mind, cartilage is like it's immune privilege as a tissue. In other words, you can take somebody else's cartilage cells or cartilage tissue, implant in somebody else without a fear of rejections. These are all peer-reviewed journal publications, and we have over 10 years of now experience in the animal and humans using our cartilage product, allogenic. So today I'm going to talk about allogenic cartilage technology and why we are so excited about it. First, about unmet medical need. All you have to do, look at the two bar graph on the bottom of these slides. Whether you talk about back pain or arthritis, the two most common causes of disability in adult. More people lose work, stay in the bed because either they got a back pain or arthritis. So huge medical needs. What is interesting about these two indications, if you really think about it, the cartilage on the knee, the cause of arthritis, is a joint disease. And for most part, when you have a back pain, probably mostly related to degeneration of your disc, which is also a cartilaginous tissue. So we're talking about joint diseases and cartilage destruction. In the case of spine, mostly is a chronic. In the case of arthritis, of course, a lot of sport-related injuries. Now, what is that we bring to the table? We work what we call with the juvenile cartilage cells. These are allogenic donor tissue, which is we get it from hospitals. And why we're excited about it, if you look at this bar graph, the young cartilage tissues, they have a much better capacity for repair and regeneration than adult. A 3-year-old damages the cartilage, goes ahead, perfectly heals that cartilage in the knee. Whereas, unfortunately, you and I, if we damage our cartilage, there's a very, very good chance with time that actually worsen and you end up with the osteoarthritic knee. So we work with the juvenile. The younger, the better. In fact, the technology, the cells that we use typically come from a year or less older donors that we collect. Now, there aren't that many of those donors around, that's true. But what the company has done over years is to be able to, once we get these donor tissue, isolate the cells, be able to expand these cells, actually maintain their phenotype, their bioactivity while we're expanding. So there are few donors that we get, we can treat few thousands of patients. So it's really a platform that we have developed. So when we get a donor tissue, what we create a cell bank. Out of cell bank, actually we can do two things. On the far right of this slide, we can actually grow a piece of cartilage. No scaffold. You just lay down these cells in a conditioned medium. They actually lay down their own cartilage tissue. Then you take that cartilage, in the case of Rivoflex, ship it to the hospitals, and the surgeons will take it and cut it, glue it to the defect. Or in the case of the back pain, degenerative disc, we actually don't do that. We take the cells, we put them in a protein carrier, in this case fibrin, and then the cells gets injected, and these cells then, as fibrin degrades, these cells actually lay down their own cartilaginous tissue. Okay, so we do have a platform technology. That's one big advantage working with the allogeneic that you don't have with autologous. There are a number of other advantages to come with that, but those are a very big one. And these are two big indications that I'm talking about, but our technology is really good. It can be applied to any joint, any cartilage disease. It doesn't have to be knee, could be a hip, could be a shoulder, could be ankle. In the case of Rivoflex, they said it's a scaffold-free cartilage. I believe we are the only company can do that. We grow a piece of cartilage without the need of a scaffold in a serum-free. It's articular cartilage shown here. It gets for damaged tissue. Of course, our targeting is sport medicine. One of the things we have done over years, we actually have developed a scalable, low maintenance manufacturing. It's a small scale, but it can easily scale up. It's everything we do, by the way. If you had the gentleman Mo this morning talking about FDA issue, we don't use serum in any of our processing. It's all a closed system. You lay down the cells, you go away 45 days later, you come back, you actually harvest your graft. We are in phase 3 trial with this product. Again, the market is 500,000 roughly arthroscopic procedures that we are targeting. Our competitor today, standard of care today, is a microfracture for most part, and our clinical trial is designed to compare our superiority of our Rivoflex relative to the microfracture. We now have a 5 years follow-up from our phase 1, phase 2. On the bottom shows actually the graph is put on the left. You look in the middle is a year later through the arthroscopy. And if you look at through the biopsy, these are hyaline cartilage. That means they're functional, good cartilage, as opposed to microfracture, for example, which only buys you a year or two, is a fibrocartilage. Ours is a hyaline, is a fiber. And really, it almost looks like a 10-year-old cartilaginous tissue. So we tell sometimes our patient, the cartilage you get is better than the one you already have, and your own is better than your own cartilage, because it is young cartilage tissue. It's just some of the data from phase 1, phase 2 trial. It's albeit it's a small number of patients, but mainly is focused on the safety. But if you look at some of the efficacy data, their 5 years follow-up, really take one patient we had did not responded. The patient did not follow exactly the physical therapy, but that's off. But usually you really get 100% healing for most 90% of the patients that we treated. You look at, in particular, I want to point out sport recreation. People used to jog or run and all that. For most part, they're back to do what they do after 5 years. Data looks, or quality of life looks pretty darn good. This is a percent of, you look at with MRI, for most part, 90% of these patients actually had 100% feeling. As compared to the some of the data we had in literature for the microfracture, which is a standard of care, we are significantly better than the microfracture. When we went back to FDA to do the phase 3 trial, they asked us, well, we had used the autologous fiber, and they said we need you to use the, we want to use the off-the-shelf fibrin. So we did a small study. This is another 14 patient, so albeit it's only 1 year follow-up. Even at 1 year, we see with a limited number of patients, it was a randomized study against the microfracture, but mostly are Revoflex treated patients. We are still, even at as early as 1 year, we show superiority over microfracture by most CUS data. All right, why we like what we do, as I said, we have a platform technology. It's allogeneic, scalable. I'll talk about that. It's a better technology. Not only it is scalable, but it's a better technology. It's a patient-friendly. You don't need to do 2 procedures to isolate the cells from patients, send it back. It's actually off the shelf. It's a cost-effective. When I say cost-effective relative to autologous, we're talking in order of magnitude difference between the cost of growing our cells versus the allogeneic. A typical donor, we expect to be able to grow 5,000 grafts as opposed to treat 1 patient at a time. It's an off-the-shelf solution we have. For this particular product, we have at least 2 months of shelf life. In other words, the graft put in a jar, send it to the hospitals. There's a 2 months room temperature. You can take it up when you need it. Again, it's a platform. While we focus on 2 biggest applications, in knee and back pain, but don't forget there's a hip, there's shoulder, there's a foot and ankle, and all the other opportunities. Hopefully later on we're going to explore. With that, let me switch to talk a little bit about our NuQ, the back pain product we developed for back pain. They're the same cells, by the way, except it's not a graft. So the cells get injected under 2D fluoroscopy. Patient gets up 5 minutes later, goes home. With this product, of course, there is no shelf life issue. Cells are frozen. We can keep them frozen for 18 months or so, at least. It's for minimally invasive outpatient. There is no rehab with this product, and we're just about to complete a phase 2 trial. Just to give you an idea who we treat, we're not going to go after like discectomy procedures. We're going after the patients that they have not responded to conservative therapy for at least 6 months, and alternative for them is surgery. Yet you look at MRI, they really don't have a significant destruction of the disc. The product, as I said, get injected, as shown on the far right, under 2D fluoroscopy, and we believe the pain comes actually from what I've shown here, what's called annulus, the outer layer of the disc. inside, the product gets injected, but actually gets penetrated everywhere in the disc. So we are targeting what we call a discogenic back pain, which is, for most part, these patients respond well to conservative therapy, but there's about 10-15% of those patients that they do not respond. There are going to be about roughly 500,000 of them are going to be subject for or a candidate for treatment with a new cure. Phase 1 trial we did 15 patients. This is a moderate lumbar disc degenerative grade 3 and Furman grade 3 and 4. And again, phase 1 we did, these patients had only 3 months of conservative therapy, at least some, believe it or not, years. They did not respond to the conservative therapy. In a phase 2, we are actually waiting 6 months to do that. We now actually have 3 years follow-up. We just correct my slide here. Again, it was mainly safety study, but we also look at statistically significant improvement on things like functional ability to lift or do whatever they normally do, and pain at 2 years later that I'm sharing with you. And it's a slide. So that was our phase 1. There is an ongoing phase 2 trial. 44 patients is randomized. We randomizing against the saline control. I'm happy to say, I believe this Wednesday we enrolled the last patient to the phase 2 trial. Should be done with enrollment. We're going to follow up for a year, and then we're going to go back to FDA and talk to them, possibly doing a SEPA trial study with them. So we're very, very, very excited. It's going well. So far, so good. We will see. We have 9 clinical sites enrolled in the United States. This is a stem cell conference, and I'm not here to say stem cells are no good, but in our situation where we had already differentiated cells, so they are one step further along than stem cells. These are cartilaginous cells. They're already programmed to produce cartilage, so there is no worry whether they turn into bone or other cell type. We prefer to use our cells. In fact, if you take our cells, inject them into the bone, they still stay cartilage. They don't turn into bone. So that's what's so exciting about it. So they are already programmed to be cartilaginous. So that's why we like working with them. There's no necessity to prime them to be cartilaginous. They're already programmed. So this is a less challenge for us than if those who work with the stem cells trying to convert them into the cartilaginous tissue. So we are one step ahead. That's with that. Thank you. I'll take any question. I got 1 minute and 44 seconds. Thanks. No, thank you. Thank you. Right. Yeah. Yeah. The chondrocyte, cartilage cells, they are immune privileged. They do not express the signal that requires for T cells to proliferate, to basically respond to rejection. There's an extensive publication on the subject when they actually lack that. In some respect, actually, chondrocytes are the immune suppressant cells, believe it or not. But they don't have that, I believe, called H7, whatever they call it, on the surface, which is comes with the other cell type that triggers the T cell proliferation, whole rejection process. So they lack that. So that's the reason for it. And be happy to share with you the publication if you like to see that. All right, honey, thank you.